Hello everyone. So today we are going to study the next point from the is the blastulation. Okay, so we are now going to continue our story of life, how the life arises from a single cell zygote to the multicellular embryo, right? So now we are turning towards the blastula formation. Okay, in our previous video we have seen the morula formation. Now, what do you mean by morula? The morula is the 16 to 32 cell stage. It contains the blastomeres. It contains the blastomeres. The number of blastomeres is about 16 to 32. Okay, so these are the blastomeres. It appears like a mulberry. So it is named as morula. So this is the morula, right? Morula. This morula, then this is the early morula. Then it turns into the late morula. How it arranged into the late morula? Just in it, the cells which are present at the periphery, okay? It having the outermost covering called as the zona pellucida, as you know. Okay, this is the zona pellucida layer. Then further on, the size remains same as you know. Okay. So what happens during late morula? The cells which are present at periphery, they become slightly elongated and arrange themselves. The small size cells which arrange themselves at the periphery. Clear? These are the cells, small size cells which are arranging themselves at the periphery, right? Then the cells which are big in size remain at inner side. Remain at inner side. So this is the early morula and this is the late morula. Okay. The formation of morula took place on the up to the fourth day of fertilization. Okay. Complete formation took place on the fourth day. Clear? So afterwards, now it turns into this late morula is now enters into the uterus. It enters into the uterus and falls from the fallopian tube into the uterine fluid, right? The uterine fluid then works on the outer layer called as this outer layer is a zona pellucida layer. As this zona pellucida avoids the wrong side implantation, clear? Okay, it doesn't allow the implantation in the fallopian tube. Very nice. So afterwards, okay, this fluid, the fluid which is present in the uterine fluid moves inside. Okay, as soon as it touches to this, the outermost layer, that is the zona pellucida layer and the plasma membrane get dissolved. Okay, it get dissolved and this outer layer cells and the inner cells get exposed to that fluid. Okay, that uterine fluid. Okay, comes in a contact with the uterine fluid. Clear? So now we are defining the blastula. I'm trying to explain the definition of the blastula. The blastula is the rearrangement of the cells of morula. Rearrangement of the cells of morula to form fluid filled cavity. To form a fluid filled cavity, blastocyst. called as a blastula. Okay, it is called as a blastula. So what is the definition? Concentrate on this diagram. Okay, so the definition says that it is the rearrangement of the cells of morula to form a fluid-filled cavity, right? It's a rearrangement of the cells of morula to form a fluid-filled cavity, okay, is called as a blastula, or the formation of blastocyst, okay, to form a blastocyst, okay, to form a blastocyst. So, how the rearrangement took place, as this is into the uterus. Clear? Here is the uterine fluid around it. Here is the uterine fluid around it. Okay. So what happened? Because of the action of the uterine fluid, this morula, okay, when enters into the uterus, the zona layer, this is the zona pellucida layer, it get dissolved or break by the action of uterine fluid, as well as the plasma membrane around it, it also get dissolved, okay, by the action of this by the action of the uterine fluid, it get dissolved. So what happened? The cells which are forming outer layer of the morula get exposed to whom? To this uterine fluid. Clear? So what happened? This fluid from every side tries to enter inside. The uterine fluid tries to enter inside. This fluid provides, especially this fluid provides nourishment to the development. Further development, it required nourishment and it is taken by this uterine fluid, and so that this fluid gives nourishment, so it is called as a uterine milk. Clear? Okay. So it having so many nutrients, and these nutrients are required for further growth, so this fluid enters inside. So what happens when the fluid enters inside? Okay, this is as the fluid enters in this, the cells which are present at the outer side get straightened. Okay, they get slightly elongated. The cells become elongated. Okay, I'm showing this change at here, blastula formation. The cells get elongated. I'm drawing it. Okay. The cells get straightened, get elongated as the fluid enters inside them. Okay. And forms the outer layer cell called as, this outer membrane is called as trophoblast. Now this straightened cells, exactly these are the outer layer cells which are of the morula, right? When the fluid get filled inside it, okay, when the fluid get filled inside it, it get straightened and arrange them at periphery. Okay, so this is the outer cells, outer layer cells, and this outer layer cells are called as a trophoblast. How the trophoblast get formed, as you know, and now the size of the embryo get changed. As you know, from the zygote up to the morula, the size of that embryo is remains same up to the morula formation, right? Then, as soon as it enters into the uterus, the uterine fluid enters inside it and it gets straightened. The cells, the pressure is exerted on the outer cells, and this cells gets elongated and straightened, right? Okay, and this layer get formed, and this layer is called as a trophoblast. This layer is called as a trophoblast. Clear, everyone? How the trophoblast layer get formed? Okay, and further on, what happened? The cells which are present at inner side, the cells which are present at inner side, they move. Okay, these cells are present at this site, right? So these cells moves towards upward direction. Why? The fluid which is exerting a pressure on these cells. Okay, and especially it moves towards the animal pole side. Okay, it moves towards this side, and here is the cells of robbers are present. Okay, here is this, especially this cells. Why they are moving at this side and why not this side? Because there are specialized cells called as the cells of robbers. Here is the cells of robbers. Okay, I'm not mentioning at here. Further on during gastrulation, you will study that. Okay, it moves towards this side, and the cells now. These are big size cells which moves towards upper side, right, towards this side. And these cells are now called as inner cell mass. Inner cell mass, forming the inner cell mass, right? And this cavity is called as a blastocyst. Okay, so focus on the definition again. Blastula is the rearrangement of cells of morula. It's just simply a rearrangement of the cells of morula. Rearrangement gets straightened, right, and moves towards upward direction and stick to one end. Okay, to form a fluid-filled cavity, blastocyst. So, unique feature of blastula is the formation of blastocyst, which contains a uterine fluid. It contains a uterine fluid, and this uterine fluid is used for the nourishment of this inner cell mass. Yeah. Okay. So this is very important point. Now we are now trying to solve our mystery: how the multicellular organism is formed from the single cell. Okay. So listen carefully. Okay. It is very interesting point, right? So. Further on, this inner cell mass are forming. This inner cell mass is responsible for formation of future embryo. It forms an embryo. From the inner cell mass, the embryo gets formed, right? And from the trophoblast, what will be the form? This trophoblast is responsible for formation of extra-embryonic membranes. Extra-embryonic material. Okay, that means especially related with this, if we see that extra-embryonic material of the membranes, material rather than material, the membranes get formed by the trophoblast. Clear? So, related with the blastula, as you know, it's simply a rearrangement of the cells of morula to form an outer layer called as a trophoblast. The cells which are present at the inner side, they move upward direction to form towards this side and glued, okay, and to form a inner cell mass and having a cavity called as a blastocyst, and thus the blastula get formed. Okay, blastula formation took place within 7 days, and further on 7 to 10 days it requires for implantation. Further on implantation. Okay, now we are turning towards the next point from this topic is the implantation. So here we complete the blastula. So take a snapshot. Okay, now we are turning towards the implantation process. Implantation and gastrulation are running simultaneously. Okay, so the next one is a sorry, gastrulation nahi. So implantation. Okay. Now, the implantation means what? This embryo, this blastula, get nourishment from the endoderm. Sorry. It takes a endometrium from the nourishment from the endometrium, right? So it gets buried inside the endoderm. It gets buried inside the endoderm. So you know the diagram. So especially I'm just drawing the uterus. Okay. So this is the uterus. It having this inner side, the endometrium, right? Okay. Now this blastula. This blastula. This blastula. Okay. And this side, here as the inner cell mass cell. Okay. So these are, especially, this is the endometrium, highly folded and highly thick and highly vascular, right? So what happens during implantation? During implantation, this blastocyst, now we can say it as a blastocyst, comes in contact with the endometrium. Okay, comes in a contact with the endometrium. Okay, so 7 to 10 days. In that, this site is called as, now this site is called as an embryonal knob site. This site is called as a embryonal knob. Embryonal knob site. And this is the opposite to it. It is abnormal. Embryonal site. Okay. So these are the two sites. So embryonal nerve site comes in contact with the endometrium. It comes in contact with the endometrium. Now, okay, see, as you know, from the inner cell mass, the embryo, exact embryo has to form, and from the outermost trophoblast layer, the extra-embryonic membranes get formed, right? So it comes in a contact with, for example, this is the endometrium. As you know, it is highly folded. Okay, these are the, yes, uterine glands. These are the uterine glands. Okay, so inner side of this, it having, okay, as you know, it having two layers. This is a stratum basalis, endometrium layer, stratum basalis, and this outer layer, which is forming the folds, called as stratum functionalis. Stratum functionalis. Functionalis. Right? These are the two layers. These are the two layers from which the endometrium get formed. And just below to the endometrium, there are the muscle layer, myometrium. So during implantation, the CET and MCQ question related with NEET. Which part participate in implantation? The endometrium and the myometrium. Some part of myometrium participate in a implantation process. Mind, especially note out. Okay, especially the stratum functionalis is the functional layer which get developed when the progesterone level increases from the corpus luteum. The corpus luteum secretes the progesterone, and so that along with the estrogen, it brings out the thickening of endometrium. This endometrium becomes more vascular and more glandular, as it having glands. Okay, uterine glands which secretes uterine fluid. Okay, and requires blood supply. And this blood supply is necessary to form further process. Okay, now we are going to start with that point. Okay, so this is the uterine glands, and here is the blood supply. These are the blood vessels. Now I am showing these are the blood vessels which are present in a endometrium, right? These are the blood vessels. Clear? So I am mentioning it. And this is the endometrium. As this embryo get implanted, then and then we can say that that female is conceived. Okay, that means that female is pregnant. When we can say it? When this embryo, this blastocyst get buried inside the endometrium, then and then only we can say that the female is conceived. Clear? So what happened? This layer is called as a trophoblast layer. Trophoblast layer. Clear? Okay, I'm showing this scenario. Okay, inside the uterus, so what happens? I'm trying to show it. This is the trophoblast. This trophoblast cells get converted into two parts. This trophoblast cells get converted into two types. The one is a syncytotrophoblast, and the another one is a cytotrophoblast. Okay, so what these two, especially the trophoblast, get converted into two parts: syncytotrophoblast and cytotrophoblast. So what is the difference between these two? The syncytotrophoblast. Okay, for example, see, here is the trophoblast. Especially this trophoblast at the embryonal knob site. Okay, this starts to divide, and to form a multinucleated membrane structure. These are the multinucleated structure. This is the multinucleated part. Okay, not so well defined. Okay, it is called as a syncytotrophoblast. And the trophoblast cells which are defined one and which remains at this side, these are the cytotrophoblast cells. Okay, so might be you find out the difference between these two. Those trophoblastic cells remains as it is, named as a cytotrophoblast, and those which are leaving this and having nucleated, these are called as the cytotropoblastic cells, which are forming cytotropoblastic villi, which are further forming cytotropoblastic villus or villi, right? Okay. Then what happened? This is the endometrial lining. See, this is the endometrial lining. These are the blood vessels inside it. It is highly vascular now. Okay, now what happened? Okay, during implantation, this change happens at this site, embryonal knob site. Okay, what happened? The syncytotropoblastic cells get formed. This thin cytotrophoblastic cells tries to innervate. Okay, they enters inside. They digest, they release lytic enzymes, and because of these lytic enzymes, they penetrate, they makes a gap or pit inside the endometrium, and it tries to moving. It continues its moving towards upward direction into the endometrium. What happened? The cells, the thin cytotrophoblastic cells, try to digest this endometrium. They break this endometrium and makes a pit. Okay, गड्ढा किया उन्होंने यहाँ पे endometrium के अंदर. So क्या? So further on, this pit gets formed, and then this whole assembly moves inside. Whole assembly moves inside. Okay, this moves inside. I'm showing from this side only. Okay. Suppose. Now what happened? It enters inside. It moves inner side. Okay. Now I'm showing the thickened endometrium, and it moves inner side. Okay, it digests this endometrium. That tries to digest this endometrium. उसको डाइजेस्ट किया, उसके बाद उन्होंने उसमें गड्ढा किया, गड्ढे के अंदर गए, बाद में ये जो रप्चर एंडोमेट्रियम है वो वापस उन्होंने क्या किया, फॉर्म किया. So totally this blastocyst get embedded or inserted or get inside into the endometrium, and this term is called as a, yes, now it is called as a implantation. Clear, everyone? So it is so easy fact. So what happened, that morula comes into the, yes, uterus. Outer events, see the events. The outer membrane get dissolved by the action of uterine fluid. The outer cell, the uterine fluid enters into the morula, so that the blastula get formed, a cavity, a fluid-filled cavity get formed, and the cells which are present, which are big size cells, moves towards the one side and get adhered to that point. That is a firm attachment now, called as a inner cell mass. These cells are called as inner cell mass, and the outer cell gets taken and called as a trophoblast. Clear? Further on, now the implantation. During implantation, what happened? This layer, this site is called as embryonal knob site, and the embryonal knob site has an ability to adhere on the wall of endometrium. Clear? So this embryonal knob site has an ability to adhere on the endometrium, right? Then when it touches to the endometrium, the change happens to this layer. The trophoblastic cells convert themselves into two different types: syncytiotrophoblastic cell and a cytotrophoblastic cell. Those cells which are moving called as a syncytiotrophoblastic cells, and those remains at that site called as a cytotrophoblastic cells. Clear? This यानी कि one type of cell वो जगह छोड़ते हैं और ऊपर जाते हैं, goes inside the endometrium by forming a vellus. They are forming the vellus, okay, and moves into the endometrium, digests that endometrium and tries to make a pit inside it, and so that they are making a cavity for its seat of the blastocyst. Clear? So that the blastocyst can sit inside it. And whatever the small damage they have done, that is repaired further on it, so that this blastocyst get embedded in a endometrium. Clear? Now, पूरा आपके ध्यान में आया? Okay, now we are moving towards further point. Then what happened further on? The syncytiotrophoblastic cells then touches to this. And what is this? This is the, yes, blood. These are the blood vessels of the mother, of the endometrium, right? And that is from the mother. Okay, so this is the mother blood. Now, these inside trophoblastic cells comes in a contact with the blood vessels of a mother, and now from which now it is not in a uterus, it is into the endometrium, and from which it get nourishment. Then it get nourishment from the blood of the mother. Now from this side, it start to take the nutrition from the mother's blood. Clear? It takes the nutrition from the mother's blood for its further development. Clear? So every individual having is grown from its every single drop of mother. They absorb the nutrient and they form the whole body. What we have that is taken from our mother's blood, and we are forming our bone, even though our teeth also having our mother's calcium. Clear, everyone? So all that formed in our body, we have our mother inside it in the form of bones also. Okay, so everyone is getting the point. Okay, further on, and this is called as implantation. Now we are turning towards the next point called as gastrulation. So the story will continue, right? So we are going to study the gastrulation. So you can take a snapshot of this point. Okay, now we are turning towards the gastrulation. So what do you mean by gastrulation? Now, gastrulation. During gastrulation, the formation of germ layers takes place. During gastrulation, the formation of germ layers takes place. And so many biochemical changes as well as physiological and morphological changes happens in this, right? So what happens during this? Okay, now we are showing the changes during or the events during gastrulation, and we have to mention it. During gastrulation, first change happens, that is the formation of embryonic disc. How the embryonic disc get formed. So this is all happens inside within this endometrium now. Okay, so we are turning towards the next point, that is the gastrulation. So during gastrulation, what happened? The embryonal disc, right? That the embryonal disc, as you know. So during gastrulation, concentrate, huh? Everyone, please concentrate in every word. So this outer layer cells are called as, yes everyone, these cells are called as a trophoblast cells. These cells are called as a trophoblast cells. So जो ब्लास्टुला है वो आगे बनके इम्प्लांट होने के बाद उसमें जो चेंजेस हो गए उसको हम बोलेंगे gastrulation. So this trophoblastic cells during gastrulation, what happens? It having the inner cell mass at this side. These are the inner mass cell. This inner mass cell now gets straightened. Look, and become columnar. These cells become straightened and become columnar. Just elongated हो जाते हैं वो cells, get elongated. Clear? Okay. And some cells become cuboidal. Here I'm showing the another cells. And some cells become, especially the cuboidal. Some become columnar and some become elongated. Okay. So it forms. This is called as an embryonal disc. This is called as a formation of the first stage. First stage. Formation of, during gastrulation, formation of embryonal disc. Embryonal disc. Formation of embryonal disc took place. Clear? So what happened? Some cells get elongated, get straightened, and some becomes a cuboidal cells. Some are becoming cuboidal cells. Those cells which are become elongated called as the epiblast cells. Just change हो रहा है cell in cells में. Okay, the cells which are elongated उनको बोलेंगे हम epiblast cell, and the cells which become cuboidal उसको बोलेंगे हम hypoblast cell. So exactly ये जो cells है ये these are from the trophoblast, derived from the trophoblast, as you know. Okay, so these cells are called as epiblast cell and these cells are called as a hypoblast cell. Epi means at the top and the hypo means at the below, right? So epiblast and hypoblast cells, and these cells are, this is called as a embryonal disc. This is simply the formation of this epiblast and hypoblast. Simply यहाँ पे लिखूंगी मैं formation of epiblast and hypoblast. So during formation of embryonal disc, what get formed? These two layers get formed. One is the epiblast and one is the hypoblast. The big columnar cells are called as the epiblast cell, and the small cuboidal cells are called as a hypoblast cell. Okay. So यहाँ पे तो cavity है, उसको बोलेंगे हम blastocell, right? So everyone is clear? Now, this is the first event, and the second event then. This is the first event during gastrulation, formation of embryonal disc. Formation of embryonal disc. यहाँ पे formation of embryonal disc. Disc form किया. Formation of embryonal disc. Yeah. Okay. Further on, the next point. Disc form होने के बाद क्या होता है? So the second point is the formation of amniotic cavity. यहाँ पे second step रहेगा formation of, during gastrulation, right? Formation of amniotic cavity. Formation of amniotic cavity. Formation of amniotic cavity. So the second step. So how the amniotic cavity gets formed, right? I'm showing the event. This is the first one. Now the second one. How the amniotic cavity gets formed? So these cells pulls downward. These cells get pulled downward, right? And to form a cavity, and this cavity is called as a amniotic cavity. Okay. Downward pull होता है उसको, and they moves downward to form a cavity. So these are the trophoblastic cells. So the events can be asked during gastrulation. Okay. These are the, yes, again, name it, trophoblastic cells, right? These are the trophoblast layer. Then what happened? A downward movement happened, and because of the downward movement, this one is the hypoblast cells. जो नीचे की तरफ होते हैं उनको बोलते हैं हम hypoblast cells, and जो ऊपर की तरफ रहते हैं उनको बोलेंगे हम epiblast. So these are the hypoblast cells. It comes along with the hypoblast. These are the hypoblast cells. So these are the hypoblasts. And here is the cavity formed. And this cavity is now named as a amnion. Amniotic cavity. Amniotic cavity. So this cavity is called as a amniotic cavity. Just what happened? This embryonal disc moves downward. It moves downward. So because of the movement of downward, a space is created between the trophoblast and this cells. Okay, and this cavity is now named as a amniotic cavity. Clear? Now the amniotic cavity get formed, right? The amniotic cavity get formed, which having the trophoblast cells at its top side. Okay, now these tropoblastic cells, which are present at this knob side, embryonal knob side, start dividing. They start dividing to form. They are forming another type of cells. And these cells are called as amniogenic cells. Now, समझ में आया क्या हुआ? Tropoblastic cell की जो which are forming the roof of amniotic cavity, these are dividing, and this dividing cells are forming another type of cell called as a amniogenic cells. So, what is the function of this amniogenic cells? कुछ तो जरूरत होगी, इसीलिए तो उनका फॉर्मेशन हुआ. So what is the need? So as the amniotic cavity get formed, it must be responsible for formation of amnion. So amniogenic cell further क्या form करेंगे? It forms another membrane. It is forming extra-embryonic membrane named as a amnion. It is forming a amnion, and this amnion is responsible for formation of amniotic fluid, and this amniotic fluid is responsible or necessary for the development of the embryo. Okay, यहाँ तक समझ में आया? आगे जाएंगे हम. So everyone, first step during gastrulation, that was the formation of embryonal disc. How it get formed? Okay, a pull happens towards downward direction. Sorry, the cells get elongated. Okay, to form a epiblast cell, and one remains at lower side called as a hypoblast cell. Clear? Okay, these two are forming bilaminar disc. Embryonal disc. This is called as a bilaminar embryonal disc. Bilaminar means two layer wala, right? It having two layer wala disc, and so it is called as a bilaminar disc. Top में रहेंगे उसमें epiblast cell, and below it is a hypoblast cell. Yeah. Okay. Further on, the second step is the formation of amniotic cavity. Sorry, formation of amniotic cavity. कैसे बनती है वो cavity? So just these cells are moving downward as this cells, the tropoblastic cells at the root side or at the top side start dividing. They are forming amniogenic cells. Okay, so this cells moves downward and a cavity gets formed. This cavity is called as amniotic cavity, and the roof of amniotic cavity. Now you can see that. The question can be asked in a CD. The roof of amniotic cavity is formed by which type of cells? Name that cells. These cells are called as the amniogenic cells, and these are responsible for formation of amnion. Okay, and this amnion is responsible for the formation of amniotic fluid, and this fluid is responsible for providing nourishment to the embryo. Clear? यहाँ तक clear हुआ? बाद में the third step. We are moving towards the next point. Okay. So ये सारा कहाँ पे हो रहा है? Into the endometrium. Okay. During implantation and gastrulation go simultaneously. These two events are took simultaneously. Okay. So the next one. Next, the third, During third, extra-embryonic xylem get formed. During the third stage, the formation of extra-embryonic xylem. Formation of extra-embryonic xylem. Formation of extra-embryonic xylem. So, extra-embryonic xylem formation. During this third stage, what happened? ये जो hypoplastic cells, this hypoplastic cells starts dividing. These hypoplastic cells start dividing. This is the epiplastic cell. Yes, these are the epiblast, and these are the hypoblast. Okay. So extra-embryonic xylem formation. Okay. During this, what happened? So, what is this layer? Yes, outermost boundary called as a, yes everyone, this is the trophoblast. This is the trophoblast. Clear? So, trophoblast layer, you have to show. This layer is a trophoblastic layer, right? Here is, yes, these cells are called as, these are big sized columnar cells. Yes, everyone, try to answer it. These are called as a epiblast cells, right? And below to it, there are the another type of cells called as a, these cells are called as, yes everyone, answer it, फटाफट answer दे दो, इसको बोलेंगे हम hypoblast cells. Okay. So what happens during this formation of extra-embryonic xylem? Okay. So यहाँ पर especially this hypoblast cells. Hypoblast cells, and this is the epiblast cell. Okay, these hypoblast cells are responsible for, here यहाँ पे बनेगा हमारा exactly the primitive endoderm. So exactly primitive endoderm. Now during gastrulation, I have stated earlier, during gastrulation exactly 3 germ layers formation took place. So how it get formed? So यहाँ पे तो क्या 2 type of cells, so one is the epiblast and one is the hypoblast cell. So from the hypoblast, first germ layer will be formed, that will be a primitive endoderm. So mind the नीट में भी पूछा जा सकता है ये क्वेश्चन, which germ layer is firstly formed during embryonic development? So that is the hypoblast. Which layer? Hypoblast. So from the hypoblast, the primitive endoderm get formed. The primitive endoderm get formed. Then this hypoblast cells are starting dividing. These hypoblast cells are starts dividing to form extra-embryonic endoderm. These cells, I'm showing in yellow, these are start dividing mitotically and forming boundary, inner boundary of the trophoblast layer. Okay, at the embryonal knob site. Clear? This is the embryonal knob site, and opposite to it is a embryonal nerve cell. Towards which, that means surround just to the inner lining of the trophoblast. It is forming the extra. It is especially forming the layer called as extra-embryonal endodermal cells. These are called as a extra-embryonal. This is the endoderm, right? Endodermal cells. Endodermal cells. But keep in mind why it is called as extra-embryonal. Yani ki, this extra-embryonal is not participating in formation of exact endoderm. Clear, everyone? Keep in mind, this extra-embryonal cells, okay, is not forming the endoderm. Endoderm kahan se banne wala hai? Endoderm will be formed from the hypoblast cells. It will be formed from the hypoblast cell. So because of this, only we are saying it is a primitive endoderm. That means exact endoderm of that embryo. Okay, exact endoderm of that embryo will be formed from the, yes, hypoblast. And this hypoblast cells divide and forms them around to the trophoblastic cells. They are forming. And here the cavity get formed. Here one cavity is get formed. And this cavity is now named as a yolk sac. It is called as a yolk sac. So यहाँ पे amnion बन गया. Amniotic cavity. Okay, यहाँ पे amniotic cavity बन गई. यहाँ पे yolk sac बन गया. दो cavities form हो गई. अपर वाला amniotic cavity, नीचे वाला yolk sac, and बीच में क्या है? Epiblast and hypoblast. As you know, from the hypoblast cells the exact, yes, endoderm get formed. So the question may be asked in the NEET that from which the endoderm get formed. That layer is called as a hypoblast. Clear, everyone? So further on, we are moving towards the next step, that is the formation of chorion and amnion. Next step, how the chorion and amnion get formed. Fourth one, formation of chorion and amnion. Extra-embryonic membrane, how they get formed. So, especially, a change occurs as it having two cavities. Here is the cell. Yes, these cells are, yes, amniogenic cells. Name है उनका amniogenic, यानी कि genic means synthesis वाला. So they are synthesizing amnion and amniotic fluid. Okay, inside it. So there are two cavities get formed. So might be the structure will change. So structural change may happen, right? And what is the structural change? This whole assembly become straightened from both the sides. And now the shape will change. They become quite elongated. It become elongated. Okay, it becomes elongated. Yes, again यहाँ का outermost layer रहेगा trophoblast. Trophoblast. Okay. ऊपर की cavity रहेगी. Yes, it will be a amniotic cavity. Okay. Here will be the amniotic cavity. Here will be this one yolk sac. And here will be अपन के रहेंगे यहाँ पे epiblast cells and another type of cell called as a hypoblast cells. These two type of cells will be here. Amniotic cavity, yolk sac, and these are the epiblastic cell, and these are the hypoplastic cell. And यहाँ पे one more get synthesized, that is, here is the extra-embryonic mesoderm gets synthesized from this side. And this is also formed from the trophoblast cell. Extra-embryonic mesodermal cells get formed. Okay. Extra-embryonic mesodermal cells get formed at this side. Okay. Further on, we will focus on this formation of amnion and chorion in our next video. Okay. We will stop here now.